So I will be talking about the BioImage Archive implementation of the REMBI model. So these are some nice images from the data sets that we already have in the BioImage Archive. But without the metadata, basically these are just that nice images. So unless you are the ones producing them, but even so, we don't know what they are, what is the organism that is being imaged here, under what conditions, how the specimen has been obtained, and so on, which are all the necessary information to make sense of those images and to potentially reuse them or reproduce them, which are the basic, principles of science. So for that, basically, the bioimaging community came together and came up with the REMBI, which is the recommended metadata for biological images, which are basically a set of guidelines for metadata to enable fair sharing of the scientific data for the biological images. And we have implemented this REMBI model, these REMBI guidelines, into the BioImage Archive. So REMBI has this 8 high-level metadata sort of containers. Study is the highest level metadata. This is just describing your project, your data set, including the funding and the publication information. The study component is basically actually a container which hosts the biosample, specimen, and image acquisition information about your images, about your data sets, and helps you to organize your project into based on the experiment types or the sample types, because one project can contain different experiments or different samples, basically. The biosample is, as in the name, it's basically the species of the organism that you're imaging. If you're imaging a particular cell line or genetic backgrounds and so on. Specimen is the actual thing that you image. It's the how you prepare your sample, how you obtain your sample that you image. And the image acquisition contains the metadata about how your images were captured, the methods the imaging method that you use, the settings, and so on. And then comes the image data, which is the image level metadata. This one is implemented in what we call the file list in the BioImage Archive, which lists all the files, all the images, and any additional non-image files that you submit. So you put the image level metadata in the file list as opposed to as a form field like the other bits. And then there is the image correlation and analyzed data as optional fields, which can be relevant to only a subset of the studies, basically. And image correlation is basically the correlation of the images obtained from different modalities, and analyzed data contains the image analysis metadata. So how we implement this is, as I said, like these form fields plus the file list. And this structure is sort of good to learn and think about in advance, because it is also how you would organize your data before submission. The simplest way is a one study component submission, where you have just one biosample and have one specimen, one sort of specimen from that biosample, and a single imaging method where you obtain your image data and potentially analyze data. But there are many different types of experiments and projects that doesn't fit this single study component. For instance, the high content screening data, where you have multiple biosamples and multiple specimens, in which case we recommend using still the single study component to encapsulate these high content screening data, but instead of like creating multiple biosample subfields, putting this information into the file list, as I mentioned, because it is basically different for from image to image. Whereas if you have a more like simplistic multi-component structure, whereas for instance, if you have multiple modalities, for instance, if you have one biosample, but you obtain two different specimens and you have two different acquisition methods, say, you know, you use some sort of fluorescence microscopy in this one and some sort of fish experiment to analyze, to image the other specimen from the same biosample, it's better to have two different study components, even though the biosample is the same. Or you can have cases where you have two different biosamples in the same specimen, like the host virus interaction, which then goes and imaged in with the same method and settings, in which case it's better to use a single study component. However, for instance, again, if you have the multimodal imaging, like I was giving an example, but if those images are then related to each other, if there is an image correlation between them, which then you later use, it is sometimes better to put them into the single same study component and capture that image correlation information that goes together with them. So these are all now implemented in the BioImage Archive. And when we go back to the nice images I show in the beginning, here you can see like they're all from, as Matthew was saying, this is a slightly different version than the Matthew showed with from two different data sets here, I think. They're from different types of organisms and so on. But like if the title doesn't give you all the information you need, but here, as Matthew was showing, we have all that information about how your sample, this is the one that belongs to this data set, the ESPIAT 605, which is the whole slide images of kidney tissues to image the to study renal cell carcinoma. Basically, you know, this contains all the information about how the sample was prepared, what is the imaging method, the settings, and so on. Similarly for this one, and all the information about our REMBI implementation is listed under the REMBI help tab here, with a reference model reference documentation as well, which lists all the individual REMBI fields that we use with some specific examples as well as descriptions.